So now we're going to look into the next major component of food that we want to measure, which is fat or lipid analysis. So performing lipid analysis is also very important for us to know what it means by lipid. So lipids are a group of substances that in general are soluble in ether, chloroform, and other organic solvent, but they are insoluble in water. We can further classify lipid according to its general classes. Okay, lipid can be simple lipid that include fat, waxes, esters of fatty acid, triglyceride, and di and monoglyceride. Lipid can also be compound lipids such as cerebrosides, sphingolipids, and phospholipids. Or when we talk about lipid, also covered on the derived lipids that include the free fatty acids, long-chain alcohol, hydrocarbons, and fat-soluble vitamins, which is vitamin A, D, E, and K. Foods generally contain many types of lipid, but the one that is most prominent will be the triacylglycerol, as well as the phospholipid. So liquid triacylglycerol at room temperature we call this as oil, whereas solid triacylglycerol at room temperature are termed as fat. Generally, triacylglycerol from animal tend to be in solid form. Interested in lipid analysis, this is important for accurate nutritional labeling and also to determine whether the food meets the standard of identity, and lastly to ensure that the product meets manufacturing specification. So prior to lipid analysis, there are a few general considerations. We know that lipids are soluble in organic solvent and insoluble in water. Therefore, this water insolubility of lipid is the essential analytical properties that used as the basis for the separation of lipids from proteins, water, and carbohydrates in food. There is a wide range of relative hydrophobicity of different lipids that makes selection of a single universal solvent impossible for lipid extraction of food. So because some lipid in food, they are part of complex lipoproteins or liposaccharides, and by having this complex structure, it increases the polarity of lipid. Therefore, in order for us to analyze this lipid, we have to successfully extract, okay, by breaking down the bond that is formed between these lipids and proteins or carbohydrates, so that this lipid can be free and further solubilized in the extracting organic solvent. There are different methods of how you can analyze total lipid, okay, which is by organic solvent extraction methods or by alkaline or acid hydrolysis, okay, followed by Montagnier extraction. So accuracy of direct solvent extraction methods, okay, without prior acid or alkali hydrolysis greatly depends on the solubility of lipids in the solvent used, as well as the ability to separate the content determined with another solvent of different polarity. So we have here three different methods of lipid extraction, okay, either through solvent extraction methods or non-solvent wet extraction methods or instrumental methods. And just to summarize the different analytical methods that we can use for lipid analysis, we can further divide them into three, either solvent extraction methods, non-solvent wet extraction methods, and instrument. For solvent extraction methods, it may be either continuous, semi-continuous, discontinuous, elevated pressure or temperature, as well as gas chromatography methods to determine fatty acid. Okay, so this semi-continuous oxalate is the most commonly used methods in most labs to determine crude fat content. Okay, Montagnier will be second. We also have the supercritical fluid extraction, but then this process is more of a green technology because we don't use most of this toxic solvent, right? But the extraction methods will be more expensive compared to the other methods here. Okay. We also, for this solvent extraction methods is suitable for semi-solid or solid food product. But for liquid sample, you may want to go for this non-solvent wet extraction methods, and this in particular is very useful to measure fat content in milk product, okay, especially for Babcock and Gerber, and instrumental methods such as infrared and nuclear magnetic resonance spectroscopy method can be used to analyze lipid, okay, but it's not commonly used for routine analysis. So prior to lipid analysis, it's very important to prepare your sample, okay, because the validity of the fat analysis of the food depends heavily on proper sampling and preservation of the sample prior to analysis. It is ideal for your sample to be, the properties of your sample to be as close as possible to the properties of the population from which it is taken. Okay, so sample prep for lipid analysis depends on the type of food as well as the nature of lipids in the food. Okay, so for example, if you want to extract lipid from liquid milk, methods that you're going to use will be different than if you want to extract lipid from solid soybean. For best result, when you prepare your sample for lipid analysis, you should carry out them in inert atmosphere or nitrogen, and as well as at low temperature in order to minimize any chemical reactions, particularly for lipid oxidation. Unfortunately, there's no single standard methods to extract all kinds of lipid. Again, it depends on your knowledge of the structure, chemistry, and the occurrence of the principal lipid classes and their constituents, okay, in the food. Okay, prior to this lipid analysis, there's some important step involved for the sample preparation. First is to pre-dry a sample, because we know that solvent, this non-polar solvent that we use, cannot easily penetrate the moist food tissue due to the hydrophobicity of the solvent used. So what you can do is either go for this vacuum oven drying at low temperature, or to use freeze drying methods to increase the surface area of the sample for better lipid extraction. Here, pre-drying helps breaking down the fat water emulsion, free fat, and thus make these fats dissolve easily. Second is to reduce the particle size by finely ground your sample. Okay, extraction of fat from finished product can be a challenge based on the ingredient, so it's best for this product to be ground after freezing with liquid nitrogen. Here, a significant portion of lipids in food, such as dairy, bread, flour, and animal product, is bound to protein. So it's important for you to break down this interaction by performing acid hydrolysis. Okay, so acid hydrolysis can break both covalently and ionically bound lipid, and thus this free lipid can easily then be extracted into in the non-polar solvent. So lastly, you want to select your best solvent. So ideally for your solvent, you want them to have high solvent power for lipid, okay, as well as low solvent power for non-lipid. You want the solvent to leave no residues, evaporate easily at low heat of vaporization. It has low boiling point, non-flammable, non-toxic, okay, low surface tension with food, single compound, cheap, and non-hygroscopic. Unfortunately, in real world, this is impossible to find solvent that fit all of these ideal criteria. So in most cases for food analysis, we use these two different types of solvent. These are the most commonly used solvent, either ethyl ether or petroleum ether. Ethyl ether is commonly used in lab, but the problem with ethyl ether is that it is very flammable. It's considered as explosive hazard. It can form peroxide, expensive, and also hygroscopic. So an alternative to ethyl ether is petroleum ether. It is an excellent solvent for lipid because it's more selective for more hydrophobic lipids. It's less hygroscopic, less flammable, and it's relatively cheaper than the ethyl ether. Different methods of lipid analysis. One is the continuous solvent, which is goldfish methods. Here is the solvent from a boiling flask will continuously flow over the sample that is held in thimble, as shown in this figure here. Okay, fat content is then measured by measuring the weight loss of the sample. Okay, once the fat being removed. Okay, so the faster and more efficient this continuous solvent extraction method is faster and more efficient than semi-continuous extraction methods, but the problem is that this channeling of the solvent can occur where the solvent may take different routes through the sample, so you may not be able to fully extract your fat from your food component, from your food sample. Semi-continuous solvent extraction, solid methods is the most commonly used method in our lab. Okay, so if you look here in this diagram, okay, you have solvent that built up in the extraction chamber, okay, and that's surrounding the sample and then siphoned back into the boiling flask. So here the fat content is measured by weight loss of the sample or by weight of the fat removed. This will then provide soaking effect of the sample, but at the same time it doesn't cause any channeling. But the problem of this semi-continuous solvent extraction method is that it will require more time compared to the goldfish method. Another good thing is that the lipid will remain in solvent due to its low volatility. Next, we look at this discontinued solvent extraction methods. This is also one of the official methods, Mojonia methods, which also involve the alkaline hydrolysis. Okay, it is this in the AOAC methods 989.05 to measure fat in milk. So here what happens is that the ammonium hydroxide is added to precipitate milk protein, and ethanol is then added to prevent gel formation. So then the fat is then extracted using a mixture of ethyl ether and petroleum ether in a Mojonia flask, as shown in this figure on the right. Okay, so this is Mojonia flask. Now this extracted fat is then dried to a constant weight and then expressed as percentage fat by weight. Okay, the advantage of this Mojonia method is that it doesn't require removal of moisture from the sample, and you can apply both liquid and solid samples. But as I mentioned before, in proximate analysis, just measuring crude fat content is insufficient. Okay, you still have to measure total fat by gas chromatography. Okay, so fat and fatty acids are first extracted from food by hydrolytic methods. This can be done by acidic hydrolysis for most product or alkaline hydrolysis for dairy product. Or for cheese, you can combine both of these acidic and alkaline hydrolysis. So in this gas chromatography methods, triacylglyceride is added as internal standard, and fat is then extracted into ether and methylated to fatty acid methyl ester. We call this as FAMES, using boron trifluoride in methanol. So now we can measure total fat, okay, by summing the individual fatty acid expressed as triglyceride equivalents. Okay, sorry. Okay, next we're going to look at the non-solvent wet extraction methods. Okay, so these are also an AOAC methods to measure milk fat. Okay, so Babcock methods. So the figures that you see on the right is the Babcock milk test bottles. Okay, so in Babcock bottle here, okay, you add sulfuric acid to a known amount of milk. Okay, the sulfuric acid will then start to digest protein, generate heat, and then release the fat. Okay, what you need to do next is to centrifuge and hot water addition to isolate fat for quantification in the graduation portion of the test bottle. This fat is then measured volumetrically, and the result will be expressed as percentage fat by weight. So Babcock is the most common method for milk fat. Okay, it takes only 45 minutes to run. Okay, so duplicate test should agree in the range of 0.1%. The only disadvantages of Babcock is that it doesn't determine the phospholipid, and it's not applicable to products containing chocolate or added sugar without modification, because of charring by the sulfuric acid. Gravimetric standard methods for milk fat analysis. This is the same as Babcock, but in Gravimetric methods, it also use amyl alcohol. So this isoamyl alcohol help prevent charring of sugar by heat and also sulfuric acid. However, it's quite difficult to read the fat content from graduated flasks, but generally for Gerber method is a much faster and simpler method compared to the Babcock method. We also have instrumental methods that is considered rapid, non-destructive, and only require minimal sample preparation and chemical consumption, such as that nuclear magnetic resonance spectroscopy, or these infrared methods and specific gravity, the force-led methods. But the problem is that these instruments are very expensive. Okay, it's expensive to run this, so it's not suitable to use these instrumental methods for regular or routine analysis. For research, you may want to utilize these methods because it can be highly specific. Okay, it can also profile the fatty acids in your sample. So now we're going to look at protein analysis. Why is it important to analyze protein? Because it is also a requirement for the nutritional labeling. But in this case, you need to analyze protein using official methods of analysis. As we mentioned before, for protein, it should be Kjeldahl and Dumas. Okay, it's also important to measure protein as quality control. Okay, and you may also want to fractionate and purify your proteins. Okay, and then you want to analyze whether the protein of interest is there and what are the physical-chemical properties of these proteins, these purified proteins. You also want to investigate the functional properties of proteins as well as biological activity of proteins. Okay, but then again, in this lecture, I'm going to keep it very brief and simple. I'm not going to cover on the protein purification or to look or to profile proteins. I will be focusing more on measuring the concentration of proteins in sample. So here we have a list of methods that can be used for protein analysis. These two, Kjeldahl and Dumas methods, is an official methods. Okay, we have AOAC methods for this, and we use these two official methods to determine crude protein for the purpose of food labeling. Okay, at the same time, you also have these non-destructive methods such as infrared spectroscopy, and we also have here methods, okay, to determine soluble protein or to determine protein based on absorbance. Okay, but then again, for these methods from URAT to these ultraviolet absorption methods, the sample need to undergo sample preparation first. You have to extract the protein and increase its solubility before you can proceed with these types of analysis. So for Kjeldahl and Dumas, what you actually measure is the nitrogen content. Okay. For the burette, Lowry dye binding methods, the zinconinic acid and ultraviolet to 80 nanometer, you are actually measuring the absorbance of the protein, okay, that bind to either dye or base or the aromatic side chain of the amino acid that absorb the ultraviolet at this 280 nanometer wavelength. For Kjeldahl methods, these are the idea is that the protein and other organic components in sample are digested with sulfuric acid in the presence of catalyst, and now this total organic nitrogen, okay, from proteins is converted to ammonium sulfide, and the digest is then neutralized with alkali such as sodium hydroxide, and then distilled into a boric acid solution. So they sporate an ion, then form, and later titrated with standardized acid, which then convert nitrogen in the sample. So the result of the analysis represent the crude protein content of the food, since nitrogen also comes from non-protein component. So here is an overview of Kjeldahl methods, starting from sample preparation and weighing, and then go to step 1, where the sample is being digested, and then step 2, 3, and 4, where sample is neutralized, distilled, and titrated, and lastly is involve calculation of the nitrogen, percent of nitrogen, and also conversion to percentage of protein. Okay, now we're going to look at overview of what Kjeldahl method, the principle behind Kjeldahl methods. As I mentioned, it can be divided into 5 different steps. So the early stage, the first step is the digestions, where the food is digested by the addition of sulfite acid and catalase. Leads to the formation of this non-volatile ammonium sulfite. Right, so during the digestions, the nitrogen is free to form this ammonium ion. Okay, and then these acids, this sulfuric acids then further oxidizes organic matters, and combined with the ammonia form, the carbon and hydrogen elements are then converted into this carbon dioxide and water. Okay, now the digested mixture is further neutralized by the addition of sodium hydroxide, okay, to up the ammonia. Okay, so this ammonia then is distilled into boric acid. Solution that contain indicator is either methylene blue or methylene red. Now you have this boric anion, which is in proportion to the amount of the nitrogen, is then titrated, okay, with standardized acid. So standardized acid here, it can be either sulfuric acid or hydrochloric acid. In most cases, we normally use sulfuric acid. So we titrate this borate anion with known normality of the standard acid. Okay, in experiment, we normally use around 0.05 N normality of the sulfuric acid. Right, so once this borate anion form are titrated with the standardized acid, this will then convert nitrogen in the sample. Okay, so the volume of the standardized acid used to convert nitrogen in the sample, okay, will be measured and will be used to calculate the percentage of nitrogen. So next, once we have this information, we further calculate the percentage of nitrogen by timing the normality of the standard acid with the corrected acid volume per gram of the sample times by 14 gram of nitrogen per mole times by 100 per thousand. So this N standard acid is normality of the standard acid in mole per liter. The corrected acid volume is the mean standard acid for sample deducted with the volume of standard acid for blank. And why we use 14 here, this is the atomic weight for nitrogen. Okay, so now a factor, in order to calculate the percentage of proteins, we can use a factor to convert percentage of nitrogen into percentage of crude protein. Because we know majority of protein contains around 16% of nitrogen, so conversion factor for this is 100 divided by 16, which is equivalent to 6.25. So to calculate percentage of proteins, you have to times the percentage of nitrogen with the conversion factor. So bear in mind, although we use 6.25 as the standard conversion factors, but some proteins, they may have different protein conversion factors. For example, rice will have significantly higher, so not significantly, but it has higher percentage of nitrogen compared to egg and meat. Another alternative to Kjeldahl method is to use Dumas. So this is also measuring nitrogen. So Dumas is a nitrogen combustion method where nitrogen is released upon combustion of the sample. The nitrogen is measured, sometimes measured by gas chromatography using thermal conductivity detector. These methods give fast result, can get results in 4 minutes. So here just to show you a general component of Dumas nitrogen analyzer, you have your sample that is combusted in the incinerator and then further reduced, undergo this copper reduction unit to convert nitrogen oxide to nitrogen, and then this will then be online to the gas chromatography and detect as a spectral spectra. Dumas is an alternative to Kjeldahl methods and it's suitable for all food type. It's particularly an official methods to determine proteins in meat, okay, such as AOAC methods 992.5 and also cereal grain, which is AOAC methods 992.23. The advantage of Dumas is that it doesn't require any hazardous chemical compared to Kjeldahl. It can be accomplished in 3 minutes, and due to recent automated instrument, you can analyze many samples at once without requiring your attention. Okay, so this automated system. Problem is that Dumas is quite expensive to be conducted because it requires this expensive instrument, and it's not highly specific for proteins because it also measures total organic nitrogen. Okay, as I mentioned before, we also have these other methods that can estimate protein content. Okay, and these methods allows you to quantify the protein content. Okay, by absorbance. Okay, there are many of these methods. They may have different principle, but the idea is what you're going to measure is the absorbance. Changes in color indicate concentration of protein. So the stronger the signal, the more intense the color produced, okay, the more proteins, the higher the concentration of proteins in the sample. Okay, but in this particular case, I'm just going to talk about Lowry assay. So Lowry is one of the common methods where it combines the burette reaction with the reduction of the falling Kjeldahl phenol reagent, okay, by the tyrosine and tryptophan residues that can be found in proteins. So here you have proteins that form a complex with copper ion, okay, releasing this hydroxide ion, okay, leads to the formation of protein and copper complex. This complex then react with the falling Kjeldahl, okay, leads to the release of hydroxide group, okay, and then form this colored protein complex with this falling Kjeldahl. Okay, so upon this interaction will lead to the formation of this bluish colored product, okay, that you can read at 750 nanometer, okay, for the high sensitivity, with high sensitivity for low protein concentration, or you can measure at 500 nanometer for low sensitivity for high protein concentration. The good thing about Lowry is that it's highly sensitive. It is at least 50 to 100 times more sensitive than burette and 10 to 20 times more sensitive than 250 nanometer UV absorption methods. It has similar sensitivity to nestlerization, but more convenient to be performed. It's less affected by turbidity of the sample and is specific than most of the other methods. It's relatively simple. It can be done within 1 to 1.5 hours. But the problem with this Lowry procedure is that it requires careful standardization. The color varies with different proteins to a greater extent than the burette methods. Okay, color is not strictly proportional to protein concentration. There are some reactions. The reactions can be interfered by other interfering agents, okay, varying degrees of sucrose, lipid, phosphate buffer, monosaccharides, and hexoamine. If in your food sample contain high concentration of reducing sugar, or ammonium sulfide and supply the compound, this is going to interfere with the reaction. So you have to take this into consideration. So there are also some special consideration when it comes to protein analysis, that what will be the selection of methods should be based on sensitivity, accuracy, and reproducibility, as well as physicochemical properties of the food material. Okay, food processing methods may reduce the extractability of protein for analysis because it increased aggregation of proteins. Okay, selection of appropriate standards or calibration is required for Kjeldahl methods. And you also need to take into consideration that in the food sample, there may also be non-protein nitrogen. Okay, so this will overestimate protein if you analyze using Kjeldahl and Dumas methods. Okay, so protein efficiency ratio could be underestimated if substantial amount of non-protein nitrogen is present in food. So last but not least, I'm going to briefly talk about the carbohydrate analysis. I'm not going to talk too much about this carbohydrate analysis. Here, just to show you what the importance of carbohydrate analysis is important for standard of identity, for food quality, nutritional labeling, for detection of adulteration, as well as for food processing. So I'm not going to cover so much on measurement of reducing sugar. I'll be focusing more on measuring dietary fiber. So what is dietary fiber? These are edible part of plant or analogous carbohydrate that are resistant to digestion and absorption in the human small intestine, and with complete or partial fermentation in the large intestine. So dietary fibers also include polysaccharides, oligosaccharides, lignin, and associated plant substance. Whereas for the analogous carbohydrate is a carbohydrate-based food ingredient that are non-digestible and non-absorbable, but are not natural plant component, for example, wax. For dietary fibers, components can be made up of cell wall polysaccharide. Okay, in cell wall polysaccharide, dietary fibers can come in the form of cellulose, hemicellulose, pectin. For the non-cell wall polysaccharide, it can be hydrocolloid. And for lignin, these are non-carbohydrate polymer that consists around 40 phenol unit. There are two ways of how you can determine fiber, either by gravimetric methods or chemical methods. I forgot to define what is gravimetric methods. Okay, so when we talk about gravimetric methods, means that we are depending on the weight of your is based on weight of the sample. Okay, so that's how you determine the component. So for both of these methods, these chemical and gravimetric methods, starch in particular can affect the analysis. So it is essential to remove all digestible starch to accurately measure fiber, and the incomplete removal can increase the residual weight and cause high value. So you will tend to overestimate your fiber. Okay, so what you can do is you can first hydrolyze starch with enzyme glucosidases, such as alpha amylase and amyloglucosidase. For gravimetric methods, first you need to digest all the starch, lipid, and protein. Okay, and then selectively solubilized by chemicals or enzyme, and then indigestible material will then be collected by filtration, and the fiber residues is quantified by gravimetrically. Here, we also include resistant starch. So for gravimetric methods is method to measure crude fiber. So crude fiber will give an estimate of indigestible fiber in food. It can be determined by extracting with sulfuric acid or sodium hydroxide. So the insoluble residue is then collected by filtration, dried, weighed, and ash to correct for mineral contamination of the fiber. Here with gravimetric methods, crude fiber can only measure variable amounts of cellulose and lignin, but then for hemicellulose, pectin, and hydrocolloid, which are solubilized, cannot be detected using this gravimetric. We also have chemical methods that can be used to measure fiber content. So carbohydrates are first removed by enzymatic digestions. The fiber components are then hydrolyzed by acid, and monosaccharides and lignin presents are then. Okay, Common chemical methods used to measure fiber is Anglis Cumming methods. In principle, the starch is first gelatinized and enzymatically digested. Okay, so the remaining polysaccharide then hydrolyzed in sulfuric acid to liberate free monosaccharide, and this neutral sugar measured by GC and uranic acid will then determine calorimetrically. But then resistant starch and lignin are not included as a component of fiber. For this method is good test for most food, but problem is it doesn't include lignin and resistant starch. So if we compare between methods, so Anglis Cumming give lowest value because lignin and resistant starch are not included. Both methods of this AOAC and this Anglis Cumming include proteolysis that cause some of the fiber to be solubilized. This will then reduce the amount of lignin, but for resistant starch, it can be measured in the AOAC procedures, but this Anglis Cumming method is slightly more reproducible. Yeah, as I mentioned early part of the lecture, okay, for Proximate, we can determine carbohydrate by difference, where once we have determined the ash, protein, fat, fibers, and moisture of the food, you can subtract the total weight of the food, and the difference is considered as carbohydrate. Okay, as mentioned before, why we calculate by difference is because there's no standard methods that be able to measure all diverse carbohydrates component. Okay, therefore, to find the percentage of carbohydrates, you just have to deduct the weight of the samples with the percentage of moisture, ash, fat, and protein. Okay, as well as if you have fiber, then you can also include that in the calculation as well. Okay, from this Proximate, you can also use this information to calculate energy. Okay, where protein in grams, okay, you have to times by 4 kilocalorie, whereas for carbohydrate, you also times by 4, fat you have to times by 9, and then you total up to calculate what would be the energy in kilocalorie. You can also further convert this to kilojoule by timing the value of the kilocalorie with conversion factors here, 4.184. Okay, so that's why if you look at the nutritional labeling on the food package, okay, you will see that it will tell you what will be the energy per serving and also per 100 ml or per 100 grams. Okay, labeling is important for mandatory nutrients. Okay, so solid food will be energy, carbohydrate, protein, and fat, whereas for liquid food is energy, carbohydrate, protein, fat, and also total sugar, especially for ready to drink beverage.